welcome, welcome, welcome to our participant town hall. Again, before we get started, I just have one more thing to walk through. We're going to go through our land acknowledgement. So at CanPath, we acknowledge the traditional territories of the Mississauga of the New Credit First Nation, Anishinaabe, Wendat, Huron, and Haudenosaunee Indigenous peoples on which the CanPath National Coordinating Center at the University of Toronto now stands. The territory was subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Iroquois Confederacy and Confederacy of the Ojibwe and allied nations to peaceably share and care for the resources around the Great Lakes. With CanPath teams working across Canada from coast to coast, we also acknowledge the ancestral territories that are home to many Indigenous people from all across Turtle Island, and we are so grateful to have the opportunity to work on this land. Here is our overview for our participant town hall today. We have Dr. Philip Awadallah, our National Scientific Co-Director, on the call already. He'll be walking us through an overview of CanPath. Following Philip, we have Dr. Jennifer Brooks, our Executive Director at the National Coordinating Center for CanPath, speaking to plans for the future. Next, we have Dr. Trevor Dummer, our National Scientific Co-Director, covering environmental exposures, so the latest research on environmental exposures using CanPath and regional cohort data. Following Trevor, we have Dr. Vicky Ho. She is one of the Cardigan or Quebec cohort regional scientific co-directors covering occupational exposures. And then lastly, we have our physical activity and diet component, or covering our heal and charm studies. And that is brought to us by Dr. Rachel Murphy, who is the lead for the heal and charm studies, the lead principal investigator. And we also have Dr. Jennifer Venna, who also works on these projects, and she is the scientific director for Alberta's Tomorrow Project. After these presentations, we have a Q&A period that's moderated by Drs. Philip Awadallah and Jennifer Brooks, where we will answer the most popular pre-submitted questions. Thank you all for submitting so many questions. We've gone through and selected the most popular ones, and then we will answer as many as we can on our website after the town hall. If any new questions come up today that you didn't ask when you register, you can provide those questions in our post-event survey. Otherwise, in our Q&A period, we also have two guests from our leadership team. We have Dr. Victoria Kirsch, who is the Scientific Director of the Ontario Health Study and our National Scientific Coordinator. And we also have Dr. Megan Vanstone, who is the Scientific Director of Healthy Future SASC. So that is our outline for today. Thank you so much for tuning in again, and I will pass things off to Dr. Philip Awadallah. Take it away.
Thank you, Megan. Thank you for leading us into this, and welcome everyone to CanPath's second town hall. As Megan mentioned, I'm Philip Awadallah, the National Co-Scientific Director for CanPath. I've been with CanPath now for almost over 15 years. And I'll start things off by telling you where we're at and where we've been, and give us a justification, if you like, for why we built the CanPath cohort, and with, of course, all of your participation. So, as many of you know, the burden of chronic disease in Canada is growing. 1 in 2 Canadians will be diagnosed with cancer. 1 in 12 Canadians live with diagnosed heart disease. And 1 in 10 Canadians live with asthma or COPD. And CanPath was built to address these questions. Why are some people developing cancers? Why are some people developing heart diseases? Why are some people developing asthma or COPD, and among other diseases? So CanPath was built as a population health research platform. It is different than a clinical trial in that what we are trying to build with CanPath is a population platform that understands and captures information about all of the potential determinants associated with the development of disease. It captures information about genetics, behavior, family health history. Through our partnerships, we capture information about environment, as well as who's developing chronic diseases and who's not. CanPath is Canada's largest population health research platform. The Canadian Partnership for Tomorrow's Health has recruited well over 330,000 participants, Canadians aged 30 to 74, from all 10 provinces, and you're going to see a breakdown of that participation rate across the 10 provinces in a moment. And we try to capture information through questionnaire data. We do some capturing of information through inviting participants to a clinical assessment center in some cases, where at that time we might actually also collect a biological sample, which allows us to look at things like genomics and other factors that we can capture from blood. And with consent, we also capture information about through administrative health records through a process called data linkage. This is a breakdown of how we've recruited over 330,000 participants across Canada. And you can see here that largely this reflects the demography of the population, with some of the largest recruitment happening in Ontario, and currently some active recruitment happening in places like Saskatchewan. So if you're in Saskatchewan, you would like to take part in the CanPath activity through the Healthy Future Saskatchewan program. They are still recruiting, and right now they've already recruited 1,500 participants. Manitoba has just ended its recruitment. They've stopped at 8,767 participants as well. So those are the two programs, those two prairie provinces, Manitoba and Saskatchewan, which were our newest partners, and we're very happy to have participation from Canadians living in those provinces. Now, one of the reasons we collect population data, and we collect data in this way rather than, say, a clinical trial, is because that we believe that capturing information before people develop disease will allow us to understand how people develop these diseases. So through what we call population laboratories, we capture information longitudinally. We capture information from participants, say, at baseline, and then doing repeated follow-ups, either through questionnaires or through linkages to administrative records. We can see how people's health progresses over time. And by doing that, we'll be able to facilitate research both in a retrospective as well as a prospective way. Now, we've been in action since 2008, and this just gives you a timeline of when we began. 2008, the Canadian Partnership for Tomorrow project was launched initially as CPTP, and now it has been rebranded as CanPath. And we've been very active, of course, since 2008, in both collecting baseline information, bringing in new partners, as I mentioned, Manitoba and Saskatchewan are our most recent activities. Baseline information, as I said, started in 2008, but we've been very busy capturing follow-up information from our participants. And you're going to be hearing about that today from some of our project leads from across Canada. We were also very busy during the pandemic, during COVID-19, capturing information from questionnaires about who was getting COVID, and also getting blood samples from our participants as well to see who was developing immunity to COVID, as well as how people were responding with regard to vaccines as well. Now, this is just a quick breakdown of the kinds of information, as I've already highlighted to some extent, that we've captured. And what we've also been doing with, as I mentioned, from collecting baseline information from 330,000 participants, is capturing follow-up information as well. The follow-up information is obviously just as important as collecting that baseline information. It allows us to capture what's happened to your health information since we first saw you at baseline. It also allows us to capture information about what medications you're taking, any changes in work status or household income, any changes in terms of environmental exposures. And by exposures, we could mean where you live, where you work, or other behaviors like sleep, alcohol, and tobacco usage. All of these are obviously important factors associated with impact on health. As I mentioned before, for many of our participants, we did collect blood samples. I'm a geneticist, and from what we've been able to do with those blood samples is also begin a fairly extensive genotyping program. And what we mean by genotyping is we capture genetic markers across people's genomes. And we've just begun this activity, and we've already gotten almost 60,000 participants genotyped, majority so far from Ontario and Quebec, with some sampling from across Canada. And of course, our aim is to capture as much genotyping information as we can from the blood samples. And here you get these kinds of plots that you see on the bottom right here. Some of the kinds of things that we can capture with genotyping. With genotyping, we're going to get information like ancestry and so on, as well as information about heritable risk factors, for example, for diseases as well. So this is a great place for me to turn things over to Jennifer Brooks, our Executive Director for CanPath, who's also going to tell you about some of the other exciting activities that are ongoing at CanPath.
Yep, thanks very much, Philip. So there are a number of ongoing data collection initiatives that we have going at CanPath, and you'll hear about some of them. But one of the ones that we wanted to bring up for you today is our work related to firefighters. So it's known that firefighters have a higher rate of cancer diagnoses than the general population. And so this has become an area of research for CanPath in partnership with Health Canada and the Public Health Agency of Canada. So we already have about 800 firefighters identified already within our cohorts, and many of them have already provided blood or urine samples. But we have a new initiative happening in Saskatchewan where we have an additional $40,000 that's been committed by Health Canada to expand targeted recruitment of firefighters so we can increase the number of firefighters that we have in our population and really start to study what cancer rates look like in this at-risk population. This is particularly important as we're all familiar with the increase in forest fires and bushfires across the country during the summer season. Next slide, please. Another example of ongoing data collection is the Ontario Health Study follow-up. So besides the ongoing new recruitment that we have in the prairie provinces, OHS is engaged in their second follow-up. And already we have information from over 27,000 OHS participants, which is really fantastic. And if we look at this data, some of this early data, we can see that we have more individuals, more women who are reporting breast cancer diagnoses. We have almost 27% of people indicating that they have a mental health condition. And because this cohort is ongoing and some of our participants are getting older, we're also seeing an increase in individuals reporting being diagnosed with osteoporosis. So all of this allows us to continue to address the health of Canadians as we continue with this data collection. Now, one of our really big initiatives that is sort of ongoing right now is a change in the way that we are giving researchers access to your data. So instead of having an approved researcher submit their project for approval to CanPath, and then we send them the data they need to answer their research question, we have flipped the model so that now approved researchers have to go through our usual CanPath data access process, but instead of us sending the data to them, they are coming to the data. We are now moving to put the CanPath data into a trusted research environment, which is basically a single cloud storage place where all of the data that has been collected is put into one secure location that then requires approved researchers to log in through two-factor authentication to gain access to the data they need, as well as all the analytic tools they need to answer their research question. We are also working to train the next generation of scientists. And one of the ways that we're doing this is through what we call a synthetic data set. And what this is basically a data set that isn't the real CanPath data, isn't the real data that you provided to us, but it looks about the same. And so I know that that can sound a little bit odd, but it allows us to share this data more freely and use it for things like workshops and hackathons and data competitions. It's also used in courses, university level courses across the country. And this really allows us to provide opportunities for students to get their hands on data from a large cohort, which they typically don't get the experience in their training to be able to do. We are also working to support initiatives by the Canadian government. And you may or may not be aware, but there has been a lot of discussion about changing breast cancer screening guidelines with mixed sort of recommendations about whether screening should start at age 40 or at age 50. Now, the Canadian government identified a couple of evidence gaps, sort of places where questions that still needed to be answered before we could make a clear recommendation about when to start screening. And we are using CanPath data to address those evidence gaps, to address those questions. And many of them have to do with any sort of differences in access to treatment, frequently based on race and or ethnicity, or it could be location, rural versus urban location, any differences in treatments that are seen across these different regions or by race, and also differences in breast cancer risk factors. And so this is ongoing work that will be made public, the results of which will be made public early in the new year. So stay tuned for that. We also have an exciting ongoing initiative called the Canadian Alliance for Healthy Hearts and Minds. And this is bringing a whole new type of data into the CanPath data set, where there are almost 10,000 of you who have also provided MRIs. And these are MRIs of the brain, of the neck, of the abdomen or your stomach, and then of the heart. And we're in the process of bringing this data into our trusted research environment and linking it with all of the questionnaire data that you have provided. There is also a follow-up, so they're collecting second images, second MRIs from participants in Ontario and Quebec. And this will be a truly unique resource for researchers in Canada. Now, Philip mentioned that we're also collecting data through data linkages. And what we mean by this is that every time you interact with the healthcare system and you use your provincial health card, it creates a data point. And these data points are used to monitor the health system, right? We track things like wait times and number of individuals going to emergency departments and so on, so that we can plan and know where resources need to go. But we can also use these data for research. And we have partnered recently with an agency called the Canadian Institute for Health Information, which is an agency that tracks all of this data in order to monitor the health system. But we can use this data to look at the health of Canadians. We can look at who's being admitted to emergency departments and why. We have information on prescription drug records for those over 65. And we also have information about physician billing claims and so on, which allow us to identify new diagnoses or new conditions that individuals are being diagnosed with. And this is really a first in Canada agreement, and is something that we're still in the process of carrying out that linkage. We've also recognized that, the importance of population cohorts doesn't just stop with CanPath. There are other cohorts that exist in Canada that sort of focus on different age groups. So CLSA is the Canadian Longitudinal Study of Aging, and that focuses on questions related to healthy aging in an aging population. And there's the child cohort study, which studies, as you might guess by the name, individuals from infancy through to now they are entering their teenage years. And so we're working closely with these leaders of these cohorts so that we can work together to really study the health of Canadians from birth all the way through to healthy aging. And this is something that we're really excited about and think will be something that's really, really unique. Okay, so before we go on to the presentations of the specific topics that Megan outlined, I just want to show you the faces behind CanPath. This is a really incredible team of scientists from across the country. And the National Coordinating Center here at the University of Toronto is also the team that works really hard to make the data ready for our researchers to answer these important questions. We're funded by national and regional supporters, and we have data hosts across the country. And this is really something that is a true collaboration from both funders and scientists across the country. Okay, so now I'm going to hand it over to Dr. Trevor Dummer. Trevor?
Thanks, Jen. Great to be here. It's really nice to be a part of this. Apologies, I'm going to croak my way through this presentation. I hope my voice lasts out. Obviously, it's the beginning of the winter, and I've gone down with a cold over this weekend. But as Jen said, my name's Dr. Trevor Dummer. I'm the national co-director alongside Philip. I've had the privilege of being part of CanPath from the very beginning. I was working with Atlantic Path and may have met one or two of you as you were recruited through the Halifax Shopping Centre. So I've been a part of CanPath in different guises since 2008. I moved to the University of British Columbia about 10 or 11 years ago, and I've worked with the BC Generations Project for a while, and have now transitioned into the more national role. And as you can see from, I guess, a really long job title that I've got, my interests are really in cancer prevention. So as Philip mentioned, he's a geneticist. And across our research teams, we have a wide range of different interests. And I'm a population health scientist, but I come at it from a much more social science view. So my training is actually in health geography. So I'm really interested in the environment and how that really impacts disease, and in particular how it impacts cancer, but how we can utilize that information for cancer prevention. And CanPath really is, if I can just, that's it, thank you, moving along. So CanPath really does provide us with an opportunity to try to understand how the environment around us actually impacts our health, because it's really crucial. We all live, work and play in specific places, and places really do interact with behaviors, genetics, environmental exposures, all sorts of other things to actually impact our risk of cancer and other chronic disease. So I as a geographer as well, we actually draw together information. That's why there's a picture of some map on here with lots of different layers of information. We use mapped data to compile different information layer upon layer to try to really understand the context of how and why diseases like cancer occur, but also then how do we use that information as a focus for cancer prevention? Because the old adage is quite true. Prevention is much, much better than cure. And the graph here along the bottom shows you that actually, cancer is a really quite preventable disease. Up to 40% of all cancers could be prevented if we can put into practice some of what we already know. But it does vary by different cancer types. So cervical cancer is virtually 100% preventable through vaccine, for example. Whereas we don't know that much about prostate cancer, so it's not a particularly preventable cancer. And there's lots of different ones in between. So what we're trying to do really with CanPath is to try to understand more about how your your information you gave us about your lifestyle, about your behaviors, about your occupation, about all these other things, how that might interact with your built and natural environment that surrounds you to give us some ideas about how we might be able to move forward to prevent cancer. So we set out, obviously, you completed detailed questionnaires, a baseline. Some of you did the follow-up. As Philip mentioned, we've had an occupational history questionnaire, the COVID questionnaire. This has captured lots of really useful information. And in terms of the environment, we asked you about your sun exposure, secondhand smoke. We also collected information on occupational exposures through an occupational history. So we got some information on your environmental exposures from you. We also got some information on your individual behaviors, like your diet, physical activity. We're going to be hearing more about occupation and also about diet a bit later on. But as Jennifer said, one of the powers of CanPath is this ability to be able to link to other databases. So Jennifer talked about linking to health records and the CAIHI information to tell us about your health experiences, your encounters with the health system. But also because you gave us your address information and your postal code, we're actually able to use that to actually connect to a whole range of other databases that tell us about the environment around you. So that becomes really, really powerful. And that's the focus of my presentation. I do want to say with these types of linkages where we're using your postal code, we don't give the postal code out to researchers. Researchers don't know where you live. They don't have that level of information. What we do is take the postal code information, make those linkages to the environmental information. And then when we provide information to the researcher, they get obviously your questionnaire data, but they get the variables associated with those environmental exposures. So they don't know where you live. They don't know your home address, for example, because ensuring your privacy is crucial to what we do. So in terms of environmental linkages, we've partnered with a major group, the Canadian Urban Environmental Health Research Consortium, otherwise known as CANU, which is a nationally funded organization that has been collating a whole range of environmental data sets across Canada. And they've been using the postal code information that we have to link your participant information to these environmental databases. So I've just put a few examples on here. So for example, air pollution, we know that air pollution, forest fires, but air pollution from vehicles and stuff is a serious health hazard. So we've been collecting information on, for example, fine particulate matter, PM 2.5. Now these are this is smoke, but you can't see it. You can't see the particles. PM 2.5, it means they're less than 2.5 micrometers. So if you imagine a strand of hair, these are particles 30 times smaller than a strand of hair. So you can't see them. Issue with those is when you breathe them in, they go very, very deep into your lungs and they sit in there and they can cause inflammation and other health related problems. We've also got information, for example, on green space. So, are there parks? Are there lots of green spaces for physical activity in the neighborhood you live in, for example? We know that can impact health. In terms of other neighborhood factors, walkability. Do you live in an area where you can easily walk around? You can walk to work, you can walk to school, you can walk to the shops, or do you live in an environment where it's like if you want to go to the local shops, you have to drive? So walkability can be really important for physical activity. So CANU have collated information on neighborhood factors, things like nighttime light. We know that light pollution at night can actually impact sleep patterns, which can also impact on health. CANU have collected a whole range of other databases, and currently there's about 40 different data layers or data variables, databases, datasets connected to CanPath, available to researchers who want to explore the impact of the environment on people's health. So how are your data being used? So some of you may have seen this article in the journal Nature, or at least seen it on the news, because it actually did make international news and international news headlines. And Nature's probably the foremost scientific journal in the world. And they were looking at lung cancer in never smokers. So although we know that smoking is a major cause of lung cancer, about 30% of Canadians who develop lung cancer have actually never smoked. So there's something else going on. It's not just smoking. And this landmark paper identified that actually air pollution is possibly or quite probably driving some of this increased risk in lung cancer in never smokers. And the Cartagena or Quebec cohort contributed data is a significant landmark paper. So your data and CanPath data are being used to really contribute to some of these major questions that are being looked at in an international basis. As well, we're really keen to support training, and CanPath data, we heard a little bit about the synthetic data set, but CanPath, your real data as well, is being used by a number of trainees as part of their training and we're supporting training the next generation of scientists. So Atul and Ezra here are two PhD students who work with me at the Cancer Prevention Research Excellence Cluster at the UBC. And they're using CanPath data as part of their doctoral research. And they also collaborated on a project using the BC Generations study, looking at cancer risk, obesity, and physical activity in relation to the built environment, so linked to some of those CANU variables. So they looked at green space, and this is a map here of green space. And sort of in the Victoria area or in Vancouver Island. So you can see the brown is less green space, the green, darker green is more green space. So you can see there's quite a lot of variation across that region. Walkability from the lighter color, least walkable, to the darker purple, most walkable. You can see that there's significant variation across Metro Vancouver in terms of walkability. They also looked at air pollution. This is NO2, not PM 2.5, but similarly NO2 is related to vehicles and cars. So you can see actually the darker the brown, the higher the pollution, and it's pretty concentrated along road networks. So this study looked at those factors and looking at how it impacts obesity, physical activity, and walkability, and also cancer risks. So this bar chart just looks at the change in things like walking, MVPA, or moderate to vigorous physical activity. This would be something that gets you sweating, and the sort of exercise that's really good for you, but also how it impacts obesity. This was a project led by a colleague, Dr. Lawrence Frank. And as you can see, there was a huge 176% increase in walking and a 78% increase in moderate to vigorous physical activity in areas that are the most walkable, whereas obesity was almost 40% decreased in areas that were most walkable. So you can see there's we're showing significant impacts on physical activity and obesity in relation to walkable neighborhoods. And what's really important is obesity is a major risk factor for a number of cancers. So we can identify areas, things in the built environment we could tweak and change that might actually reduce cancer risk. Now, this bladder cancer graph is a little bit complicated. But I mean, what I would say is, if you look at this, if the dot, which is the hazard ratio, if that dot is greater than 1, there's an increased risk. If these sort of the bands with little sort of gray tails, if they don't cross 1, then you would say that was statistically significant. I've just honed in here on the bladder cancer in relation to NO2 and what a tool found in the BC Generations project, where there was actually a significant increased risk of bladder cancer in relation to NO2 exposure. So overall, we could say that in relation to bladder cancer, we didn't find much in relation to greenness. The walkability was a little bit inconsistent. I didn't go into too much detail on that, but there was potential protective effects, but it was very difficult to untangle from the impact of other environmental exposures. But the NO2 did see a significant increase in the highest category of NO2 exposure, so a significant increase in bladder cancer. In terms of physical activity and obesity, it was pretty clear that a walkable neighborhood strongly impacts and increases physical activity in the participants, including vigorous exercise, but it also decreased overweight and obesity. Just want to finish off on focusing on some work that started in Atlantic Path in Atlantic Canada. And this was something that I was involved in from very early stages, and the Atlantic Path toenail collection part of our study. So all of you from Atlantic Path will know the Atlantic Path as the toenail study. We were the only regional cohort to actually collect toenails. What's really important or really useful about the toenails is once you clip them, they're about 8 to 12 months old. So things that accumulate in them, which includes environmental heavy metals, have been around for a long time. So that they are a really good measure of a long-term exposure. So really good indicator potentially of long-term risk, because we know that certain heavy metals, environmental heavy metals, and other environmental contaminants such as arsenic do impact cancer. I would be remiss not to mention as well that this is the subject of the Guinness World Record for the largest toenail collection, and there's been lots and lots of research done in this area that's been published in a number of really major journals, Environment International, Science of the Total Environment, where we found some really novel things about arsenic in the toenails. I won't go into too much. I'm not going to go into detail about these papers, but if anyone's interested, you can get links to them at the Atlantic Path website. You can also get links to them on CanPath's website as well. But it's not just necessarily about looking at arsenic and toenails and other things. There's been lots and lots of research conducted. Some of this is in Atlantic Canada. Some of this is in some of the other regional cohorts as well, looking at things like physical activity and walkability or diet quality in the neighborhood environment, how environmental factors impact sedentary behavior, chronic obstructive pulmonary disease, et cetera, et cetera. Again, not going to go into detail about these papers, just to show you that your research has really, your data has been used extensively in really high quality research that's contributing to a number of important areas. And these areas are all useful if we're looking at cancer prevention, because they're identifying environmental risk factors. Finally, just want to talk a little bit more about some of the more recent toenail research. So we had a study funded by Prostate Cancer Canada, led by a team at Dalhousie University, that was looking at the relationship between toenail arsenic and prostate cancer. And we've now expanded that to look at all cancers. So basically, you take the toenails, and probably one of the worst technician's jobs available is the technician has to open these little bags of thousands of little toenail clippings, put them into a device that can digest them and break them down into something that can then go into a vial, that can go into a machine that's pictured here, which can actually measure not just arsenic, but about 22 to 24 different environmental contaminants. And so we can actually build up a really detailed picture of the types of things you've been exposed to. And that's got real potential for cancer prevention, because we can identify in the toenails potentially biomarkers that might be able to be linked some way to a cancer sometime in the future. Potentially, they could be a risk factor for the cancer. So if we can identify biomarkers in toenails of exposures or combinations of exposures that you have actually had, we might be able to identify people that will be ideal for early detection, for screening, or for preventative types of precision medicine. And this is particularly exciting, this analysis, because it's using a branch of artificial intelligence called machine learning to really try and make sense of, you've got a suite of 22 to 24 different environmental heavy metals. How do you try to understand how some of those might be contributing to something that's going to cause cancer somewhere down the line? So this is very exciting research. I think that is me. So I'll now hand you over to Dr. Vicky Ho, who's going to tell you all about the occupational exposures.
Thank you, Trevor. Hello, everyone. My name is Vicky Ho, and I'm a professor from the University of Montreal. I'm also one of the scientific directors of the Quebec Cartesian study. I'm very happy to have the opportunity to share with you some of my work. Today, I'll be presenting on occupational exposures to endocrine disrupting chemicals and cancer risk. So let's jump right in. So what are endocrine disrupting chemicals, or I'm going to refer to them as EDCs throughout this talk? So they're a group of chemicals that can alter the proper functioning of our endocrine system, which includes the production as well as the regulation of hormones. And consequently, this disruption can cause adverse health effects, such as cancer. Now, we're all exposed to EDCs in our daily life. This could be via food, our environment, the use of certain personal care products. And given this widespread exposures, EDCs are actually really hard to study. But exposures in certain workplaces occur at much higher levels, and that's why my team focuses on the occupational environment, because it gives us this wide contrast between those who are highly exposed in certain jobs versus those who are just exposed via their daily living. So we focused on EDCs and their ability to disrupt hormones, because hormones, and particularly sex hormones like estrogens or testosterone, can play a role in the development and the progression of many cancers. My team focuses on 3 types of cancer: breast and prostate, which are the most common cancers amongst Canadian women and men, accounting for almost 1/4 of all cancer cases. As well, we also focus on colorectal cancers, as over certain age ranges, women actually have a lower risk of developing colorectal cancer than men, and these age ranges appear synonymous or maps well onto the timing of the menopause transition. So our study was funded by the Canadian Institutes of Health Research to investigate whether those who are exposed to EDCs at their workplace were at a higher risk of developing breast, prostate, and colorectal cancer. The main hypothesis was that exposure to EDCs at high doses, like those in certain work environments, can disrupt sex hormone levels and thus contribute to cancer risk. And this association may be different between men and women, and we were particularly interested in exploring sex differences in the etiology of colorectal cancers. So to do this, we selected participants from 4 regional cohorts in CanPath that had at least 30 new cases of cancers developing. These included participants from the BC Generations Project, Alberta's Tomorrow's Project, Ontario Health Study, and Quebec's Cartagen. So as you may recall, data collection in CanPath consisted of you coming into an assessment center for physical measurements, as well as completing this baseline questionnaire. For occupational data, currently all of CanPath have self-reported their current job, as well as their longest held job. And since cancer is a chronic disease that takes a long time to develop, we used information on participants' longest held job to estimate occupational exposures, since we thought that this maps on best to the development of cancers. And we identified new cases of cancers by linking your information to the provincial cancer registries. And we used a study design called a case cohort design, which essentially compares the occupational exposures amongst participants who develop cancers in CanPath to those without cancers at the onset of CanPath. So in CanPath, now that we have information on everybody's longest held job, we were tasked with how do we evaluate your occupational exposure. So I'm just going to briefly go over 2 main approaches. First, we could use an expert assessment approach where we have experts assign occupational exposures based on the information you gave us. This approach is generally considered to be the best approach in assigning occupational exposures from the past, but it is costly, and the data quality really does depend on how much information you gave us, as well as the expertise of the experts themselves. Another approach is to use what we call a job exposure matrix, or a GEM. And a GEM is essentially just a fixed set of rules to associate a list of possible exposures to a given job title or a given job. And one of its main advantages is that it's cheap and quick to apply, though its validity does depend on the type of GEM you choose to use, and that everybody who's assigned this, who has the same job total, would be assigned the same level of exposure. In our CanPath analysis, we weighed these strengths and weaknesses and decided that the application of a GEM was most pertinent. So we used a GEM called the Canadian Job Exposure Matrix, or CanGEM, to estimate occupational exposures to 17 EDCs that's listed here at the bottom of the slide. Using CanGEM, we classified participants into unexposed, possibly exposed, or exposed to any of these 17 EDCs. We were also interested in whether exposures to certain EDCs that affect estrogen or androgens differently would have a different impact. So we also classified participants into unexposed, possibly exposed, exposed, according to the way each EDCs interacted with hormones. So in other words, they can be estrogenic, or they potentiate the effect of estrogens, or anti-estrogenic or anti-androgenic, where they antagonize or decrease the effect of estrogens and androgens. So here are the results to exposure to any of our 17 selected EDCs and breast cancer risk. I know it's a bit complicated, so I'm just going to take the time to walk you through the figure. First, there is the arrow pointing to the vertical line, which indicates no association between exposure to EDCs and breast cancer risk. Everything to the right of the line would indicate a higher risk. Everything to the left of the line would indicate a decreased risk. Results here are shown by cohort, but if we pool all the results together in CanPath, I'm going to direct your attention to the red box. So here, if I interpret this box for you, for all of the participants in CanPath, we see that participants that were possibly exposed to any of our 17 EDCs experienced a 16% increased risk of breast cancer. In other words, that little diamond is to the right of the line, indicating a higher risk. While on the bottom graph, we see no association. In other words, women that were exposed to any of the 17 EDCs had no increase in breast cancer risk, and this is because the diamond touched that vertical line of no association. I'll walk you through another set of results, just shown another way, again, for breast cancer. Here I'm showing the odds ratio, which is just a statistic that's used to quantify the association between being exposed to EDCs and breast cancer risk. When I compare participants that were possibly exposed or exposed to any of the 17 EDCs against people who were unexposed. So this table shows you the same results as I shown previously for any exposure to the 17 EDCs, as well as those by different modes of action. So to interpret these estimates, anything greater than 1 would indicate a higher risk of breast cancer. So again, drawing your attention to this red box, what we see is a consistent increase in breast cancer risk amongst those who are possibly exposed to EDCs, and this is independent on how they affect sex hormone action. It seems to increase breast cancer risk regardless. Now, if I move on to our results for prostate cancer, the interpretation of these graphs are the same as before as for breast cancer. Again, if I draw your attention to the red box, what we're seeing here is those who are possibly or exposed to any of our 17 EDCs were not associated with prostate cancer risk compared to those never exposed, because again, these diamonds are crossing my vertical line. Same results shown in the table for any of the 17 EDCs and prostate cancer risk, but I do want to draw your attention to the red box, where what we're seeing here is an increase in prostate cancer risk among those who are possibly or exposed to EDCs that potentiate the effect of estrogen when we compare it to those who are never exposed to estrogenic EDCs. I'm going to conclude with our results for colorectal cancer, and here I'm just showing in this first set of table all men and women data combined. And what we're seeing here is no association between exposure to any of our 17 EDCs and colorectal cancer risk, although the risk estimates are close to 1. However, if we look at people that are exposed to EDCs that antagonize the effect of estrogens, we see an increase in colorectal cancer risk. As I said, I was interested in whether the associations were different in men versus women. So here I'm showing you the results by sex. Across the board for women, we actually see no association between exposure to EDCs and colorectal cancer risk, while a very different story emerges for men, those who are exposed to any of our ADCs or EDCs that antagonize the effect of estrogens and androgens were at a higher risk of developing colorectal cancers. So in the interest of time today, I only showed you grouped results. I didn't show you the results by individual EDCs. And here I'm just highlighting very quickly in blue for the EDCs that were common to both breast and prostate cancers, whereas the EDCs highlighted in pink were the EDCs that were only associated with increased breast cancer risk. In green was increased prostate cancer risk. The associations with colorectal cancers behaved a little bit differently. Arsenic and another agent called polychlorinated biphenyls were associated with colorectal cancer risk in both men and women, while lead and mercury was specific to men only, and trichloroethylene were specific to women only. So I will conclude with a few important considerations when I interpret the findings that we see in CanPath. First, overall, the prevalence of exposure to EDCs were actually fairly low across the CanPath cohorts, which limited our statistical power to estimate some of our associations. And second, I think I've already alluded to this a little bit. There is a degree of misclassification that's to be expected when we use a tool like a GEM. people that hold the same job title are assigned the same level of exposure, even though they may be exposed to different degrees. And we're not able to capture this, with a gem. And third, we did see differences across cohorts. In other words, sometimes what we saw in Quebec wasn't necessarily what we saw in Alberta, and we don't have an explanation as to why we see this. But just to sum up, overall, we see evidence that exposure to EDCs in workplace contribute to the etiology of breast, prostate, and colorectal cancers. And a potential biologic explanation is their ability to disrupt sex hormone levels. We are continuing our analysis for lung cancer now, and we hope to be able to share our findings with you in the future. So before I pass it off to Rachel and Jen, I would just like to thank you for your participation as well as your ongoing commitment to CanPath. Thank you.
Good morning or good afternoon, depending on where you're joining from. It's my great pleasure to present today alongside my colleague and my dear friend, Dr. Jennifer Venna. I'm Rachel Murphy. I'm originally from Ontario, and I obtained my PhD in nutrition from the University of Alberta. I'm now an associate professor in the School of Population and Public Health at the University of British Columbia, and I'm a scientist at BC Cancer. And I am leading the Heal and Charm studies along with a great team of investigators and staff and trainees from across Canada. And I'll just briefly introduce my co-presenter, Dr. Jennifer Venna, before I kick things over to her. Dr. Venna is the scientific director of Alberta's Tomorrow Project and a research scientist at Cancer Control Alberta and Alberta Health Services, and one of the leads of Heal as well as Charm.
Perfect, thanks, Rachel. And Rachel also humbly didn't mention that she was the main driving force between getting the funding for the studies that we're going to talk about now. So we're very grateful to have Rachel on board and leading this charge in CanPath with myself and Dr. Sharon Kirkpatrick, who's at the University of Waterloo. So I think as the previous studies have alluded to, as a CanPath participant and as a participant in your regional study, you really have the power to change the future. Your data is already contributing to a number of amazing studies across the country. And we're going to tell you about how you have the opportunity to do that again and continue to do that with diet and activity data. So we're going to talk about why we want to hear all about what you eat, what you do during your day, including everything: sleeping, working, volunteering, taking care of your house, your yard, and your kids. So I just want you to take a minute to think about what you've eaten so far today and what you've done. And then I want you to think about why you've done those things. Like, what has helped shape what you chose for breakfast this morning and the activities that you've done so far in your day. So for example, I too have a little bit of cold, just like Trevor, and my 3.5-year-old son decided to wake up at 5 in the morning, which is 2 hours earlier than normal. So that definitely affected my morning so far, and will probably impact my day going forward. Because it affected my sleep. I'm probably going to be hungrier. I'm probably going to be less inclined to be reaching for the carrots and broccoli for a snack, for example, and more likely to reach for another coffee. So there's those sort of closer things that can impact what we do, like our health and our personal preferences, our normal routines. But there's also broader factors that can influence things that we eat, the foods that we choose, and the activities that we do, and things that we don't maybe even really realize, like the types of marketing and media that we're exposed to, and the environments in which we live and work, and where we choose to spend our time. So the HEAL study, or the healthy environments for, what is it again? Healthy eating and supportive environment study. I talk about this study all the time. We're specifically interested in the environment. Both the physical built environment, what you would normally think of when you think about your neighborhood, right? Like the options where you have to be able to walk, maybe spend time in a park or a green space, the type of transit that you have or facilities around you. And then also more specifically, what we call the food environment. So this is where you can get access to food. This could include your garden in your backyard or your home, but it can also include like the convenience store, how many grocery stores and what they have available to you. So for example, we have a couple of grocery stores that are reasonably close to us. If it's a nice day on the weekend and I have time, I'll take my son in our little wagon. We'll go for a nice walk to the grocery store. We'll pick up our food and go home and make supper. But if there's a snowstorm in Alberta, or if I'm busy on a weekday, I'm going to choose to drive there instead. So a lot of things will affect how we access food and what is available to us. Excuse me. So as Trevor mentioned, a significant number of cancers could be prevented at the population level, or there's a lot of things that you can do to reduce your risk at the individual level. And part of this is through eating well, being physically active, having a healthy body weight, and not smoking. And often the health guidance really focuses on what you as the individual can do to eat healthy and move more. But we know that there's all these other structural and system factors that influence the choices that we are able to make. So by studying the broader environment, we can better understand how structures and policies and programs can really help support people to make the healthy choice, the easy choice, to help with your individual risk and help prevent cancers and chronic diseases on the population level for hopefully generations to come. So I think as Vicky and Trevor really beautifully highlighted, Canada is actually one of the world leaders in measuring aspects of the environment, both from the sort of pollution level, as well as the structures. Like Trevor mentioned, the Canoe Project, which will map out access to green space and walkability and things like that. But where we're really lacking is very, very good data on what Canadians eat. So most surveys will only ask you about the fruits and vegetables you eat, which is great. It's a marker of a healthy diet, but then that's really all you can talk about. There's nothing else about the rest of the diet, which also might be important. So some of the regional studies in CanPath, like Alberta, Quebec, and the Atlantic provinces, we have asked you before more detailed information, but honestly, this was quite a long time ago now, and we know that what you eat can change over time for various reasons. And then also, if you just think about how the food available in grocery stores and elsewhere has changed over the last, say, 10, 20 years, it can be pretty significant. So we want to be able to capture those changes. And there's really no other data set in Canada available now that has measured at a really detailed and descriptive level what people eat and also how they move. So this is going to actually fill a major gap in Canada. And so the HEAL study is going to include all of the regional studies in CanPath from BC all the way to the Atlantic provinces. Cartagena, if you're in our Quebec cohort, you don't see your logo there, but you are participating indirectly through another study called the Nutri-Quebec study, and we are working very closely with them to make sure that we can line up as much of the data as we can. So this HEAL study is going to be the largest collection and most detailed collection of diet and physical activity data in Canada ever, as far as we know. And so we're hoping for at least 100,000 participants to take part. You'll receive an invitation for this through your regional study. And if you're in Manitoba and BC, I think Atlantic provinces, you may have already received it. So please check it out and take part. And if you haven't received anything yet, just wait because you might be getting it soon. And so what we're going to be asking you to do is fill out a brief survey about you that will collect some background information. And then we're going to ask you to fill out some surveys that will ask about what you ate and drank and all the activities you did over the previous day. And we're going to ask you to do that a few times over the course of the year, because like we said, these things can change day to day, and they can also change over time. And so if we ask you to do what you did the previous day, hopefully it's a little bit easier to remember, and you can give quite a bit of detail about, say, what you had for breakfast, where you consumed that, did you make it at home, did you have it on the go, etc. As well as how you moved, including sleeping, taking care of kids or others, volunteering, working, leisure activities, basically how you spent your day for a full 24 hours. And then approximately at the 1-year mark, we're going to ask you a different set of questions that try to capture what you did over the previous year. And this is really to help fill in gaps. So when we ask you what you did the previous day, we might do that in the spring and the fall. And that obviously leaves out the summer and winter, where we know activities can be a bit different. And so the past year questionnaire is going to try to help fill in those gaps. Okay, so now I'm going to pass this over to Rachel to talk about the CHARM study.
Thank you, Jen. As Jen mentioned, the next study we'll talk about is called CHARM. It's quite a mouthful of title, so that's why you'll hear us referring to things as CHARM and HEAL. CHARM is a sister study of HEAL, and we know that a one-size-fits-all approach to health and cancer prevention in general is limited in effectiveness. And the reason for this is because we know that people's preferences, where you live, your metabolism, they're all different. So CHARM really aims to embrace those differences in biology, genetic, environmental factors to help us understand why some people stay healthy and others go on to develop diet-related diseases, including cancer. The other really important aspect of CHARM to highlight is that CHARM is focused on achieving equitable health outcomes. And by that, I mean, we know that there are people and groups in Canada termed structurally excluded who are more likely to develop diseases like cancer and may also be more likely to face challenges with eating well and being physically active. And CHARM is intentionally inviting participants who are structurally excluded to ensure that the evidence that we generate in this study is inclusive of the diverse populations in Canada. So CHARM will include about 4,500 participants who are taking part in the HEAL study. Invitations to join the CHARM study will start to be emailed over the next few months and into the spring. And participants who are part of the BC Generations Project, Alberta's Tomorrow Project, and Manitoba's Tomorrow Project may be contacted for this. And as a participant in CHARM, you'll have already completed most of the questionnaires in HEAL. So we are not asking people to do extra diet or physical activity questionnaires. The extra piece of CHARM is that we are asking participants to provide one stool sample using a kit that would be mailed to your house or that would be provided at a local lab or study center. And we're also asking participants to provide a blood sample, which again would occur at a local lab or at a study center. The stool sample will be stored, so in the future we can measure the types and amount of bacteria in your gut, also known as the gut microbiome. And blood samples will be used to measure health markers like blood lipids, blood glucose, as well as biomarkers of dietary intake. So this is a question that I saw in some of the questions that were submitted by participants here today. So really, how are we going to be using the data that are generated in HEAL and CHARM? So along with myself and Dr. Venna and our team, we'll be answering the questions in HEAL around dietary intake and movement behavior and the environment. But the impact is really much bigger than this. So your anonymized data will be available to researchers across Canada to answer a whole range of questions around how nutrients in your diet and what type and amount and what combination can help prevent cancer. So I've just put some examples here of the types of questions that we would be able to answer with this really detailed data on diet as well as movement behavior. And then in the future, we'll be able to look at questions around how does what you eat impact your gut microbiome, for example, and how can this help us really inform evidence and policies to help people live long, healthy lives. So just I'll wrap up in the next couple of slides. I know you've been on this town hall for quite some time now, so just really want to say thank you so much for being here today, and really we are looking to you to help us meet our research goals in Heal and Charm. So our goal is really to have about 100,000 participants enrolled. So far we've reached over 3,000 participants. So we have a long ways to go, but we're really excited to be doing this work, and thank you to those of you who have already participated. And lastly, thank you so much for listening to us. If you want to learn more about the Heal and Charm studies, you can use your phone to take a photo of the QR code, which is on the slide here. This will open the Heal and the Charm web pages, or you can also find it on the CanPath website. So you can see the CanPath address there, and then you'll be able to find more information about the Heal and Charm studies. And thank you so much, and I'll turn things over to Philip.
Thank you, Rachel, and thanks, Rachel, Jen, Vicky, and Trevor for your great presentations. We're going to move now to our participant questions section of the today's webinar. We received almost 3,000 questions before this webinar, and we have managed to get them down to a few that we'll have time to answer today. The rest that we aren't able to get answered today in the webinar in the next 15 minutes or 14 minutes that we have left, we'll answer on the website. So Jen and I are going to pass off moderating today back and forth, and we're going to start with our first question. The first question goes to Megan Vanstone, who's helping lead our Saskatchewan activity. And there we're going to ask her how many participants are part of the study at the national provincial level, and how many original participants are still involved. Megan.
Yeah, hi everybody. Thanks for the question. So as you saw in our presentations a little bit earlier, we've reached over 330,000 participants nationwide. That number actually will continue to go up a little bit because Saskatchewan is still recruiting participants. So we're hoping to contribute, another 7,000 is our target in Saskatchewan. And overall, we have seen a lot of continued engagement. So we're averaging less than 1% of participants that are withdrawing from the study. And I think it's important to note that even when participants do choose to withdraw from continued follow-up or continued engagement, a lot of times they're still consenting to us retaining the information that we've collected, retaining the biosamples that we've collected. So that allows us to continue using them for future studies, even if they choose not to continue with the follow-up activities. So thank you for your engagement. We hope to continue to see those numbers throughout the years, and it helps us retain a really, really valuable data set for the country. So thanks for that.
Thanks, Megan.
All right, our next question goes to Trevor and Vicky. Are cancer rates increasing or decreasing in the Atlantic provinces? And why is there so much cancer in that region?
Okay, I can take a first stab at that. So first thing to say is sort of cancer in Canada is increasing because cancer is predominantly a disease of older age, and as the population ages, more people get cancer. So obviously we do get cancers in younger people as well. But so generally speaking, we're seeing an increase in cancer rates across Canada. That said, Atlantic Canada does have some of the highest rates of a whole range of different cancers than the rest of Canada. So, for example, in Newfoundland, they've got the highest rate of colorectal cancer, actually one of the highest rates of colorectal cancer in the world in Newfoundland itself. That's believed to be a range of different environmental and behavioral type exposures, but there's a strong association with genetics and a genetics mix within what is an island population. And island populations often see specific types of genetic sort of markers that predominate amongst that population. So there's definitely a genetic component to that. Atlantic Canada is also, if you look at lung cancer, and I mentioned this earlier, lung cancer is a disease of smoking, but there's also a large component of lung cancers occur in never smokers. Atlantic Canada has also got some of the highest rates of lung cancer, but in fact around about 30% higher, some statistics show, in Atlantic Canada compared to the rest of Canada. So why is that? It's a combination really of unfortunately smoking rates, some relatively poorer diets not always, but tend to be a bit higher in some of the Atlantic provinces. Some of the environmental exposures that we know are really associated with lung cancer, such as arsenic, radon gas, which seeps into the home, air pollution, tend to be higher in those areas as well. So there's a whole range of other factors that do unfortunately mean that we do see high rates of cancers in Atlantic Canada, which is one of the reasons why CanPath is really important, because we're capturing lots of really important information on lots of Atlantic Canadians to hopefully identify areas we can focus on prevention, because that's, as well as treating people that develop cancer, we really need to prevent.
Thanks, Trevor. And Vicky, did you want to add to that?
No, other than just to mention that the 2025 Canadian Cancer Statistics Report was actually just released yesterday. And there you can actually see a breakdown. I mean, they have lots of sort of interactive graphs where you can look at a breakdown by geographic region for more information.
Great. Thanks, Vicky and Trevor. My next question is to Jennifer Brooks and Megan Vanstone. How is it being ensured that the CanPath participant population reflects the diversity of the Canadian population? Maybe we'll start with Jen.
Yeah, this is a great question and something that we spend a lot of time thinking about at CanPath. And, most of our participants have already been recruited. And if you look and you compare our study population, so compare the people who have given data as part of being part of the CanPath cohort, we reflect what's seen in the Canadian census. So what this means is that CanPath as a population does reflect the diversity of the Canadian population. But that doesn't mean that we're not always trying to do better. And you heard from Rachel and from Jen about how we're prioritizing in some of our new recruitment efforts to make sure that our follow-up is complete and is really being emphasized to a diverse population. During our COVID-19 activities, we also prioritized ensuring that we had captured a diverse population. And, some of the ways that we do this is by recruiting through lots of different methods. And maybe Megan, who is still in the field and recruiting new participants in Saskatchewan, can talk a little bit about that.
Yeah, thank you, Jennifer. That's really the biggest challenge, is trying to find the best ways to reach the most people across the province. So early on, we really focused on establishing more of an online presence, marketing campaigns, just trying to get awareness out there about the study. But we definitely saw higher participation rates in our urban centers in Regina and Saskatoon. So for us in Saskatchewan, we're really shifting our focus towards a broader kind of recruitment strategy, doing things like direct mail campaigns to every single resident of the province. We're really excited to launch that in the new year so that we can make sure information is landing in every resident's mailbox, even if they don't have that ability to find the information online. And we're also really going to lean into a bit of a partner engagement strategy. So really trying to identify those partners within the province who have their own networks and their own groups of individuals who we can kind of tap into and make sure that we're connecting with them in the right ways. So you saw that earlier in the presentation, our firefighter recruitment efforts are one of those examples of our partner engagement strategy and how we're trying to make sure that groups that are typically underrepresented in research are captured in our national data set. So thanks for that question.
Thanks, Megan. Okay, the next question is for Vicky. Have findings from the study contributed to changes in cancer screening, diagnosis, or treatment?
Well, cancer research, you have to sort of appreciate, is fundamentally an incremental process. So, major breakthroughs don't really happen overnight. They're the results of really all of these countless smaller definitive studies, like some of the ones that you heard about today. And they each contribute a little piece to this sort of overall puzzle. But in addition to all of the work on understanding cancer etiology, we have researchers who are using CanPath data and who are focusing on a couple of really promising areas of research that might lead to improving survival rates through earlier diagnosis. So the first is looking for biomarkers for early detection. So we have some promising work underway looking at novel biomarkers. So as people develop cancer, they have these precancerous cells that could release DNA into the blood. And we're looking to sort of collecting pre-diagnostic blood samples to see if some of these biomarkers will show up in the blood years before a cancer diagnosis. So there are novel technologies that we're using to do that work. And then the second is we have funding from the Public Health Agency of Canada to look at ethnic differences in breast cancer screening. So we're really in the midst of this research to look at differences in screening uptake and effectiveness across various ethnic groups. And so we really, hope that these two areas of research will contribute to clinical practice in the near future.
Thank you, Vicki. So my next question's for Rachel. So you just touched on this in your presentation with about CHARMS and HEAL. And so the question is, what questions can expert researchers expect? What questions can we expect researchers to ask using the new HEAL and CHARM data?
Yeah, that's a great question. So we're collecting a whole lot of information on dietary intake and movement behavior. So we'll be able to ask a large range of questions. Some of the ones that we're really interested in, you've probably seen lots of news articles recently about processed foods and ultra processed foods and health. So that's something that we'll be able to look at in our study because we'll have really detailed data beyond fruit and vegetable consumption. As well, we'll be asking movement behavior, but that's inclusive of different types of activity, sedentary activity, sleep. So we'll be able to look at questions around the type and amount of time spent in different activities and how that relates to health, and over time be able to understand how those things could both potentially increase or decrease the risk of developing cancer.
Thanks, Rachel. Okay, I think this might be our last question because we're coming up on the end here, but it's a good one and it's for you, Philip. How do you plan to use AI to support research or identify disease patterns?
So AI is becoming a very exciting tool in the population health space, particularly for programs where they've collected lots and lots of information from thousands, hundreds of thousands, millions of participants. And programs like CanPath, which has collected thousands of variables from our participants as well, from imaging data to genetic data to administrative health record data. And there have already been some significant advances using AI to identify predictions using just administrative health record data from programs like the UK Biobank, administrative health records from almost the entire population of Denmark. And so those are the types of exciting things that we're hoping to apply with CanPath data, particularly when we have access to all this great environmental data that Trevor exposed us to with during his presentation. But there's a real opportunity here for us to identify specific features that can help us predict health trajectories potentially before people develop those chronic diseases. So that's something we're particularly excited about. And that also lends particular application identifying new biomarkers potentially in the cancer space potentially. And we already are using AI tools in identifying pre-diagnosis signatures. And we're doing a really good job in the prostate cancer space using CanPath biologics presently.
Awesome. Thank you, Philip. Okay, so that is where we will end things off for our CanPath Participant Town Hall. Thank you, everyone, for joining in. Thank you to our presenters. Thank you to our Q&A panelists. Thank you to all of the participants for joining in. If you happen to miss any of this, or if you would like to send any of this content to any family, friends, people in your network, it will be available on YouTube in about a week's time. You will receive a follow-up email with that link. And if you don't receive it, you will be able to just see it on our YouTube channel in a week. And we will get your questions. The most popular questions that were answered at registration will be available on our website soon. A lot of questions were asked, so please be patient, give us some time, and they will be available on our website soon. Otherwise, have a wonderful week. Thank you for starting off your week with us, and take care. Thanks, everyone.